Perlstein couldn't be here in person and sent a video, so go ahead and share that.
Hi, everyone. My name is Ethan Perlstein. I'm the founder and CEO of Perlara PBC, the world's first biotech public benefit corporation that partners with entrepreneurial groups like Cure Mito Foundation to develop medicines for rare genetic diseases. Today, I'm excited to share an update from what we call the SURF1 Cure Odyssey, specifically a yeast-powered drug repurposing project we launched 2 years ago. We're on the doorstep of the clinic with what we hope will be the first rational combination therapy for mitochondrial diseases like Leigh syndrome. The image on this slide depicts complex 4, an extraordinary assembly of proteins, cofactors, and lipid. The questions we're asking are, how did it all come together? And how do we turn that knowledge into medicine? I like to think in visual metaphors. I imagine SURF1 as a choreographer, quietly working behind the scenes to make sure the dancers hit their spots at the right times. We know SURF1 helps an iron coordinating cofactor called heme, which must be snugly slotted into the complex 4 active site just right. SURF1 collaborates with other assembly factors to ensure smooth performance. When SURF1 is missing, what can the cell do? We hypothesize that small molecules, starting with known drugs and bioactive compounds, could activate pharmacological bypasses that allow the cell to compensate for the loss of SURF1 function. What could we realistically expect a small molecule to achieve, given that the active site of complex 4 is complex? Iron and copper ions must be coordinated in 3-dimensional space by a swarm of histidine residues. The critical tyrosine residue forms an unusual crosslink with a nearby histidine that stabilizes the reactor core. Perhaps a small molecule could boost the function of other complex 4 assembly factors or protein chaperones. Maybe a small molecule could act on the surrounding membrane to facilitate complex 4 assembly, or some other mechanism we haven't dreamed of. The only way to answer unknown unknowns in biology is with an unbiased phenotypic screen. We pulled on a thread that extends back 20 years. It was under our noses the entire time. Scientists first modeled SURF1 deficiency in yeast in 2002. SURF1 is called SHY1 in yeast. Knocking out SHY1 in yeast causes failure to grow in respiratory conditions, i.e. conditions that require functional mitochondria. We could readily exploit that grow, no-grow readout in a high-throughput drug screen. No one had done that before, so we did. At Perlara, we popped up a yeast drug repurposing lab where we optimized a 3-step process. First, we make sure the yeast mutants, or avatars, are what the label says they are, and that the published literature about them checks out. Second, we determine how many yeast cells are required to run a drug repurposing screen. And third, we demonstrate that the yeast avatars can be statistically distinguished from healthy control cells. After that, it's on to the screens. Over the course of about half a year, we screened 25,000 compounds. First, a pilot screen of 2,500 compounds. Then, 2 expanded libraries that exhaustively cover the clinically actionable drug repurposing chemical space. Plotted below are Z-score distributions. The higher the Z-score, the better the rescue, and vice versa. Interestingly, compared to other diseases, SURF1 has a fair number of rescuers, but relatively few sensitizers, or compounds that make the SURF1 yeast avatar sicker. We validated dozens of hits in dose-response experiments in yeast to determine potency and reproducibility. Here's an example of a hit that rescued growth to wild-type levels, something we only observe in about 1 in 4 diseases we run through our pipeline. A high degree of rescue suggests that a pharmacological bypass, or potentially even pharmacological replacement of SURF1 is possible. That got us thinking some more. Many of the top rescuers share structural features, or pharmacophores, that are consistent with activity at the complex 4 active site. Recall the histidines and tyrosine that envelop the active site, as well as that unusual histidine-tyrosine crosslink required for complex 4 activity. You've heard of molecular glues. These would be molecular crutches. I've highlighted in green the portions of the histidine-tyrosine crosslink pharmacophore that are present in 2 of the top yeast SURF1 rescuers. One is an activated phenol that resembles tyrosine, and the other is an antifungal called voriconazole, part of a large series of azole antifungal compounds. Notably, the histidine-tyrosine pharmacophore is a doublet, repeating twice in the same molecule. Complex 4 is an off-target of these drugs, but that doesn't mean we couldn't optimize them to become SURF1 pharmacological prosthetics one day. In a stunning example of convergence between AI, neurons, and yeast, Professor Prigione's lab identified azole antifungals, like the ones we found, as complex force active site stabilizers too. Drug repurposing can be a starting point for new drug discovery. We can now imagine a suite of designer complex force stabilizers that enable the assembly to form and function in the absence of SURF1. There's a lot of mechanistic studies that need to be done to prove the complex for pharmacological prosthesis model. We need to think about how to translate our results to the clinic now, though. We tested 12 yeast hits rescuers on SURF1 patient fibroblasts in the seahorse assay. Remarkably, 6 of the 12 had some positive effect on mitochondrial bioenergetics. ATP production rates are increased, and 3 examples are shown here out of those 6 that are active. And while these are modest, 10-15% boosts, they are increases nonetheless, demonstrating the predictive value of unbiased yeast-powered phenotypic drug screen. Finally, the most compelling drug repurposing candidate to emerge so far rescues the hyperpolarization of mitochondrial membrane potential in at least one SURF1 patient fibroblast. This drug inhibits a pro-inflammatory pathway, and we believe in combination with a molecular crutch, the two would form the basis of the first rational and targeted mitochondrial cocktail. Thank you for your time. And next steps include hit validation in disease models, including combination tests of molecules that we think are activating compensation pathways, as well as molecules that we think might be acting by this pharmacological prosthesis mechanism. We need to do further mechanism of action studies, but we also want to take some of our most promising compounds as monotherapies into the clinic in what we call 1-to-N drug repurposing studies, which will enable us to actually establish benefit in a human being while also potentially validating biomarkers we could use to prove efficacy in a larger study. I want to thank the Polar Yeast team and other collaborators that helped make our screening possible, and of course, Cure Mito Foundation for sponsoring this work and for being a great partner. Thanks, everybody.